Thank you for coming to the talk. My name is Naveed Ishaque. I'm a group leader at the Berlin Institute of Health, and I'm leading a group called Computational Oncology. The name is a little bit misleading because actually we do mainly non-oncology projects now, and we have a strong focus on single cell and spatial omics modalities, especially with regards to kind of better understanding human diseases and also for methods development. And for my talk, I'm going to introduce a kind of alternative paradigm to the traditional analysis workflow. And the talk is titled Cell Segmentation Free Analysis of Spatial Transcriptomics Data. Hopefully throughout the presentation, I will give a broad overview of the different types of technologies that can generate spatial transcriptomics data. And for single molecule spatial transcriptomics data, talk about how we can adopt some new cell segmentation free modeling approaches. So this is really about analyzing spatial gene expression data without specifically knowing where the cells are. And hopefully I'll give you a couple of examples of how indeed this is possible and how some approaches have successfully implemented computational methods to do so. I'll also demonstrate some of the developments in our group. I'm particularly excited to show you a web application that was published earlier this year where we can actually analyze an in situ sequencing data set on a mobile phone. And then later on, I'll describe a new analysis framework for analyzing spot-based data called Plankton.py, and this will be the focus of the tutorial run by Sebastian in my group later on. So quickly to get you reintroduced, I'm sure I don't need to motivate this crowd with regards to the importance of spatial transcriptomics methods. So this won Nature Method of the Year back in 2020, and spatial patterning of gene expression plays an important role in normal development, as shown in this really beautiful image of the Drosophila embryo, where each one of these images shows spatially localized gene expression in a different color. And later on, this spatially resolved gene expression will result in different tissues and organs being developed in the correct places of the organism. So while these technologies are still in their infancy, we're already beginning to see application in human diseases in the clinical setting. In particular, in regards to cancer, to identify tumor heterogeneity. In this example, we can see an immunohistochemistry slide next to some cluster analysis of spatial transcriptomics data to identify different subclones within the tumor, which may result in different clinical decisions being made as a result of this heterogeneity. So with this basic motivation of the importance of resolving spatial gene expression, I'll describe several technologies that can be used to measure gene expression in space. One of the earliest methodologies for resolving spatial gene expression are microdissection-based technologies. So these identify and isolate regions of interest within a tissue. So this can be small tissue regions, or the most advanced technologies can even isolate individual cells. And then the isolated tissues or cells can be dissociated and then put forward for sequencing, either by bulk or single cell RNA sequencing. So once you get the gene expression profiles from the sequencing, you can now map this back to the tissue because you know where you isolated these different tissues from. And over here, the resolution of gene expression directly correlates to the size of the isolated regions that you have. Another very popular technique is in situ hybridization. So this is where fluorescently labeled probes for specific gene targets hybridize to RNA molecules in situ. Some of the modern implementations do not target individual genes, but they rather adopt a barcoding strategy. So this means that in combination of different hybridization events, you can decode what gene is specifically located where via the use of things such as code books. This allows you to massively upscale the number of genes that are profiled, allowing for really kind of high throughput analysis. One of the great things about in situ hybridization is sensitivity, so you can really detect very lowly abundant molecules. But this also has a negative effect, which is it can cause overcrowding. So if you target a gene that's too highly expressed, this overcrowding can kind of saturate the signal in the microscopy image, making it very difficult to isolate individual spots. Another kind of upcoming technology is in situ sequencing. So this was developed from the lab of Mats Nielsen. And compared to in situ hybridization, in situ sequencing methods determine transcripts by in situ sequencing by ligation or synthesis. A lot of these technologies are based on padlock probes by rolling circular amplification, which amplifies signal or amplifies the target molecules in situ. Compared to in situ hybridization, in situ sequencing is a much lower detection efficiency, so it's not as sensitive. But if you remember, one of the issues with in situ hybridization is that it can be too sensitive, leading to overcrowding. So one of the benefits of ISS is that you have much less overcrowding problems, so you can actually sequence much larger areas without overcrowding. Probably the most popular spatial gene expression method is through in situ capture. Some of the most popular technologies in this kind of domain include Visium, the original spatial transcriptomics slides, SlideSeq, and HTST, for example. So these methods capture transcripts in situ, but they sequence them ex situ. So since these particular methodologies don't require construction of target probe sets, they can profile transcriptome-wide gene expression patterns, and also they reduce the overall time of doing your experiment, since you don't have to allocate specific time to designing a probe set, which can effectively reduce the turnaround of analysis. One of the most interesting ways of reconstructing spatial gene expression is purely in silico methods. So this is where a computational tool will look at spatial gene expression, look at co-expression of genes, look at expression of receptors and ligands, and then computationally reconstruct the tissue that they came from non-spatial data. I was very skeptical when I first saw these methods, but when you kind of go through the papers and test the tools out yourself, you really see that there's a lot already encoded about spatial organization just in non-spatial data. And the existence of these methods really demonstrate how far we've come with our understanding of cell biology and machine learning. So this is super exciting. Over the last years, we've seen a trend in an increasing number of technologies that can capture spatial transcriptomics data. And they're generally evolving to not only capture more cells and more genes, which basically puts a big strain on the computational analysis. So there's a really big emphasis on having easy to use and efficient computational tools to make use of this this complex, highly dimensional data. So we can also describe these spatial transcriptomics methods in the context of their spatial resolution. The resolution ranges from capturing small regions of tissue via, for example, laser capture of a small tissue region, to profiling a small number of cells in situ, by in situ capture using something like a Visium array, all the way down to capturing the location of individual mRNA molecules using ISH or ISS methods. The resolution of these methods plays an immediate role in determining the initial computational challenge. For example, low resolution methods provide a readout of multiple cells within a tissue region. So in this image, you see some tissue in pink, and you have these readout spots shown in the tissue, and these individual colored dots representing cells. So you can see that in some of these spots, you get a readout just from a single cell. In some spots, you have two cells, but they're of the same type. But in other spots, you can see that there are multiple cells of different cell types, and you would need to apply some sort of deconvolution algorithm to identify what cell type signatures are contributing to the readout in this particular spot. On the other hand, high-resolution methods require that these individual mRNA molecules are aggregated into cells. So the first step in this process is actually to identify where the cell is located. So this is usually through a process called cell segmentation, which can be done through, say, for example, staining of nuclei, and then expanding the nucleus region to include the cell, and then assigning these spots based on expected gene expression. For my talk, I'm going to focus primarily on this single molecule based spatial transcriptomics methodologies, because that's what my group specializes in, and it's also very appropriate for this workshop, because in later labs we'll be analyzing this lung developmental data set that was generated using in situ sequencing. All right, so hopefully now I've given you an idea of how we can measure gene expression and some of the initial computational challenges that are presented once the data has been generated. So how do we actually go about measuring spatial gene expression within cells? So the first thing we have to do is identify locations of mRNA. So in this example image, you have a highly multiplexed image of the tissue, with each of these tiny dots representing a detected gene within the tissue. So this is something that you should have generated in the labs that you had yesterday. Downstream of that, we need to start identifying where these cell locations are through a process called segmentation. So once you've identified that you have cells in particular locations, you can start quantifying gene expression within those cells, and then afterwards assign a cell type based on the gene expression of that cell. So that first step in that process is cell segmentation. So this is essentially a classification task. So this is where you would label individual instances of cells from a microscope image. And in particular, this can kind of be broken down into two steps. The first step would be imaging a particular cell landmark, and the second step would be to adopt a computational method to identify where the cell locations are based on that particular stain. The most common staining method that's performed is DAPI staining, which stains for nuclei. This is a well-established method and is accessible to a lot of labs, but it has a major disadvantage in that it stains for nuclei and not cells. So if you, what a lot of people do is stain for the nuclei and then expand these to get a circular shaped cell, which works in a lot of cases, but for many types of cells, they don't always adhere to this simple principle of uniform circular cells. And then you would have misclassification of, say, for example, cytoplasmic mRNA. Due to this, there are some other stains that people also use, such as, example, NISL, which is a stain for neuronal cells, and it would primarily mark the body of these neuronal cells, or a stain of poly A to identify location of mRNA, which originate from within the cell. And these would give you a better representation of the cell body. What we can also do is, if you don't actually have any sort of stain, is produce what is called a max projection. So this is where you would take all of your gene expression data and combine it into one signal and assume that this is somewhat representative of a poly A stain, which should allow you to do some basic segmentation. So the advantage of using cell body staining methodologies is they give you a better representation of cells, but compared to DAPI, the problem could be that these cells now are more adjacent than what you would expect nuclei to be. So nuclei should be well isolated, so you should be able to identify the centroids of these cells quite well. Whereas if cells are touching, you might not be able to tell them so well apart using, say, for example, a cell body stain. So this is where the final type of stain, cell membranes, might also be very useful here, where you would take an alternative strategy to actually identifying the borders of these cells as a way of demarcating. So once you have these stains, you would process them computationally. The most common segmentation method is called watershed, but there are a number of other machine learning approaches that are emerging, including those adopting deep learning models underneath them. I won't go into so much detail of these methodologies because I'm going to focus on cell segmentation-free approaches for the rest of this talk. To motivate you why cell segmentation-free approaches are important, it basically goes down to this notion that cell segmentation is not easy. It's a really difficult task. To demonstrate that, I have a couple of examples of why this is not easy. Cells themselves can have very complex shapes and sizes. So even if you use the correct stain, it might not be easy to reconstruct the exact shape of the cell. In the image on the top right, this is an example of an astrocyte cell that has been isolated from a rat brain. And you can see already that this is a very complex shape, centering around the nucleus here, which is stained in blue by a DAPI. And it's also interacting, getting very close to other cells, which are also stained, or other nuclei, which are stained in blue. So even you can imagine that there would be very few computational methods that would be able to segment an astrocyte really well. So that's a biological difficulty, but there are also some other technical difficulties that could lead to segmentation being tricky. Sometimes you simply don't have very good signal from your DAPI stain. Sometimes your cells are really tightly packed and you cannot demarcate cells really easily. So in this example here, you can see within the same DAPI stain towards the left, you have some very easy to segment nuclei. But towards the middle, you see there's a big drop in signal relative to what's observed from the left. And these cells are so tightly packed, it's very difficult to actually determine where one cell starts and where one cell ends. And lastly, probably the biggest limitation is the standard stain that's used for cell segmentation is DAPI, and this only captures nuclei. So while for a lot of cases you'll be able to model a cell reasonably accurately, you can see, for example, in the case of this astrocyte, if you stained a DAPI nucleus, there's very little chance you'll capture the real complex shape of the cell itself. So you would really be missing most of the mRNA that's in the cytoplasmic region of these very complex cells. All right, so hopefully I've convinced you that segmentation is really not a trivial task. Whilst there are a lot of tools that exist, so this is an analysis step that no one seems to really be paying attention to and assume that it's solved. So with this in mind, a couple of years ago, my group, we tried to address this issue of, do we actually need to identify cells to make sense of spatial transcriptomics data? So I'll go through an example to show that this is indeed possible and that mRNA molecule organization is not random. So here are some molecules that were detected by awesome fish in the mouse brain from the group of Sten Linison. And you can see that in these three panels, we see genes that were detected in the region of tissue, separated by what cell types they should be encoding for. So here are some genes that were detected for inhibitory neurons. In the middle, some genes from excitatory neurons. And on the right, some genes that were detected for astrocytes. So with very little imagination, you can already see that there are regions in the tissue with high density of localized gene expression corresponding to markers of particular cell types. And I'm pretty sure some of you in the crowd were already thinking of like, okay, we probably have cells in these locations here. So it's not so tricky to actually come up with some very simple heuristics that would be able to identify cells based on some spatial data that would be generated. So this essentially means we can use mRNA distribution as a proxy for cell locations. So when moving forward with this, there's some basic principles that would need to be considered. So we would need to have some sort of spatial model of how these mRNA are organized when they come from the same cell, and then some sort of cell type model of how spatial gene expression is associated with different cell types. To go into a bit more detail here, whenever you are trying to model mRNA distribution, some things that would need to be considered would be what kind of model you would have for this kind of point process data, with point process data being essentially mathematical points that are identified in Euclidean space. So this is not isolated for just spatial transcriptomics, but there's an entire field dedicated towards this with spatial statistics. So there's a lot of methodologies that already exist. You would need to also consider things such as molecular densities and how molecular densities within cells versus background varies. And also you can have more complex methodologies that also consider the neighborhood of molecules. So how are molecules connected when they're within a particular cell or within a particular subcellular feature. And then once you are able to identify these recurrent spatial trends in the data, we can start thinking about cell typing. So how are spatial gene expression associated to different cell types? And for this, once we have some knowledge of prior gene expression signals, either from single cell RNA sequencing, we can start annotating them very quickly. But we've also been able to identify recurrent signatures from the tissue itself and identified cell type distribution from that way as well. So to give a quick overview of some computational tools for cell segmentation-free analysis of spatial transcriptomics data, I've listed a couple here. And a lot of these, you'll notice, are very recent, with the earliest peer-reviewed publication coming out only 2 years ago. So this was a tool called Spatially, which was developed in the group of Carolina Valby. There was a tool called BESA, which was from the group of Peter Karchenko. And for my own group, we have a number of other tools called SAM, SAMLite, and Plankton.py, which I'll tell you a little bit more about during this presentation. There's also a gap between these pure segmentation-free approaches and segmentation-based approaches, which are now being referred to as augmented cell segmentation methods. So they use gene expression data to improve upon classical segmentation. And this includes a number of tools such as BESA. So this can perform complete segmentation-free analysis, but you can also provide some sort of DAPI prior, so it works with the initial segmentation to improve it. There's a tool called PCISEC coming from Mats Nielsen's group, which adopts a point process model with negative binomial distribution. And this has been proven to be very effective for in situ sequencing. There was a tool from Dana Pier's group called Sparkle, which adopts a multivariate Gaussian distribution in terms of modeling spatial gene expression, where it assigns dangling RNA to a cell. So this would be RNAs that might have been missed in the original segmentation. And another interesting approach is called JSTA, which combines the segmentation and the cell typing in the same step. So this applies a deep learning filter on top of the classical water segmentation and iteratively improves upon classical segmentation methods. I won't focus so much on the augmented cell segmentation methods, but rather only the cell segmentation methods, giving a brief overview of the works from the groups of Carolina Valby and Peter Karchenko, and then a bit more detail on the works from my group. An important consideration here is actually these methods are being used in terms of in the context of large consortia such as SpaceTX, which developed the much adopted Starfish pipeline. So going into the first tool, Spatially. So this is a graph-based tool for identifying recurrent cell structures using graph neural networks. It's designed to be able to capture spatial structures. So normally we would be thinking in terms of spatial structures of cells, but this can also capture subcellular structures within cells. And it's a completely unsupervised method, so it doesn't take into account prior single-cell RNA-seq data. And the basic algorithm revolves around generating a spatial neighborhood graph based on the molecules and their distances from each other. It then uses a graphical neural network called GraphSage to create some representation vector of higher order spatial relationships, which are then later embedded and visualized using the UMAP algorithm. And then you can perform some unsupervised clustering to find classes of these recurrent spatial structures within your tissue. The other tool I mentioned was Bezier. So this is a graph-based tool for segmentation of cell structures using a probabilistic model of various cell attributes. It essentially fits your spot data to a KNN neighborhood composition vector, which is statistically labeled using Markov random fields, MRFs. And then these are embedded into a two-dimensional UMAP where you can perform some annotation and clustering. The basic algorithm is that it creates a global spatial neighborhood graph through Delaunay triangulation. The graph is modeled as a Markov random field where the edges between the molecules have different strengths based on their distances and their similarities. And then it will try and optimize a model, which can be optimized on various parameters, such as a model for background noise versus signal detection. There's a model that relates to the molecules within a particular cell type. So this is using single cell RNA sequencing as a prior and optimizing that. The model can also take into account prior segmentation for its initialization, so it's not a random initialization. And you can also provide an expected cell size as a prior as well. And these graph parameters and latent models get optimized using an iterative technique via the expectation maximization algorithm. All right, so now I'm going to go into a bit more details of some of the developments from my own group, starting with a tool called SAM. So this is a fairly new segmentation-free method developed by Zhong Bing, who used to be a PhD student in my group and now is a professor at Busan National University in Korea. So the core idea of SAM is that mRNA distributions directly correlate with likely cell locations. So what we do is model mRNA distribution as a Gaussian process under the central limit theorem. So this essentially translates to areas of high spatial gene expression correspond to very likely cell locations. So if we can then treat areas of high spatial gene expression as analog of single cells, we can actually continue with the standard single cell workflow, but analyzing at the pixel level rather than at the cell level. The algorithm itself can be split up into 4 basic steps. First, calculating the spatial mRNA density, then identifying the cell type signatures, and from this, generating a cell type map. And as a downstream analysis, you can identify some tissue domains. In the first step, you can calculate spatial mRNA density. And to do this, we use a kernel density estimation with the Gaussian kernel. This is a very popular adopted signal processing step where essentially you smooth the image. So this changes the detection of discrete mRNA molecules into clouds of gene expression, which would represent the original cell from which they came from. So in this particular step, there's one important parameter for us to optimize, which is the bandwidth. So this is essentially the strength of the smoothing. So if you have a bandwidth that's too small, you smooth between the molecules, but at the level of a cell, you have a very heterogeneous distribution of expression. Whereas if you smooth too much, you end up with a circular representation where you lose the original shape of the cell. Whereas if you find an optimal bandwidth, you can maintain both parameters of the cell structure and also have a uniform distribution over the cell, where you could compute that the centroid of the cell is at the point of the highest spatial gene expression here. So this KDE approach is then applied to every gene in this kind of highly multiplex image. So you have a stack of these, which are then combined to what we call a gene expression vector field. So this could be considered as a highly multispectral image, where you have multiple channels for each gene, and in that image, each pixel is encoding a gene expression value. So when stacked and combined, each pixel essentially has its own expression profile. So again, we're moving the classification task from cells to pixels. So the next step is actually to identify cell type signatures. We can do this in an unsupervised way or a supervised way. So if you already have cell type signatures from single cell RNA-seq experiments, you can already use that to start classifying the vector fields. If you don't have a gene expression signature already, you can actually very easily compute it from the image itself. And from this, what we do is we identify representative regions in the tissue, and we treat them as analogs of cells. And this is a fairly simple process. So to go through the steps, we would have our original image of mRNA locations originating from different cells, in this case, A, B, and C. We would apply the kernel density estimation to create these clouds of gene expression over the tissue. And then we would essentially look at, within this vector field, where are the locations of the highest local gene expression, which we call local maxima. So at that precise pixel, we would sample the gene expression and then put this forward to standard single cell analysis methodologies such as clustering and annotation. So this is very, if any of you have worked with single cell data, we would typically apply a clustering method. I think at the moment we're using Louvain, but we can easily switch to just the other methodologies for clustering, such as Leiden. And then we would calculate per cluster or per cell type an average gene expression profile, and then map this back to the tissue. So once we have these signatures, we can also visualize them using a UMAP, and you can quite easily see that it's very difficult to tell that this is actually a unsupervised UMAP of cell type expression from a spatial image without segmentation at all. So we were very confident that this is working correctly. Yeah, so as I said, as you have your gene expression signatures where you have different cell types with their different gene expression profiles, you can start mapping this back to the original vector field to identify pixels that correspond to individual cell types, and then start classifying the image. And from your highly multiplex vector field, which is relation to genes, we can now classify pixels in relation to the most likely cell type in which we have a signature. So actually, with regards to the classification of pixels, so this can adopt any sort of model that you're interested in. In the end, we actually settled for Pearson correlation. So this is probably the simplest way to assign a cell type to a pixel, but we found out that it worked really effectively. But this can be exchanged with any classification method that you prefer. So once we have the cell type map, we can also identify tissue domains. So for this, we used a sliding circular window over the tissue, checking the cell type composition within each window, and then performing cluster analysis, and then identifying different tissue domains. So in this cartoon image, we can see that we're reconstructing the laminar structure of the brain neocortex. So hopefully you have a better idea of like how the tool actually works. And to demonstrate how effective this is, I will show how we've generated some results of applying SAM to some mouse brain tissue using Awesome Fish. So what we found in the initial de novo cell type signature analysis, so this is actually identifying cell type signatures directly from the tissue without priors. When we compare it to known signatures, we get a very, very high correlation. And we get a nice distribution of cell types. So really having a nice cluster of excitatory neurons and inhibitory neurons. And we generally see like the glial cells clustering together as well. So we're very confident that the gene expression signatures that we recovered were in line with expectations. When we apply the classification algorithm, you can see that the SAM reconstruction of the tissue is very similar to the original publication. We tend to argue that our reconstruction is much richer. You see a much more dense representation of cells, especially with regards to this region at the bottom, the ventricle, where it's particularly poor in the original publication. Looking in closely at this region, we actually find that the original publication wasn't able to reconstruct this very well because of low DAPI and low poly A signal in this region. So this really meant that they weren't able to segment the region so well. So you can see that the poly A signal is quite low, and so is the DAPI, especially if you compare it to just underneath the ventricle. However, if you look into marker gene expression for cell types in this region, it's quite clear cut. You really see quite high localized gene expression for the markers of the cell types in this region. And you can see that the ventricle cell types that were reconstructed by SAM actually very closely match the expected cell type distribution of the markers, whereas a lot of these were missed in the original publication. We were also able to identify the laminar domains from the tissue as well. And again, this is using our simple sliding window approach, and they're highly concordant between the original publication and our analysis as well. So one of the major benefits of the tool is that this KDE can be applied easily in 3D as well as in 2D. This is particularly relevant because classical segmentation methods do not work so well in 3D. There are a handful of tools that do, but this is quite elusive for most groups. So in this turntable video, you can see 3D reconstruction of the mouse brain profiled using MERFISH. Unfortunately, the best we could do in terms of 3D was a 10 micrometer slice because it's the only data we had available. But if you look closely, you can see that there are certain clear cell features that can be reconstructed. So hopefully I've demonstrated that SAM is indeed an interesting tool for cell segmentation pre-analysis, but you're probably wondering how tricky it is to use. So unfortunately, it's a standard Python package, which means you have to be good at managing your Conda environments, knowing how to use Python on the command line, and having access to some reasonably good hardware. To make the tool more accessible, Sebastian in my group developed the evolution of the tool called Lite, which essentially tries to minimize all of the complexity of the algorithm to make it easy to compute, and provides this on an easy to use web application framework. So the tool itself is available on GitHub, and you can test it out on the web app on Netlify. The tool itself is really light. We managed to compute, I think, around 10 million spots in just under a minute using less than 2 gigabytes of memory. And for very small data sets, we can even run it on a mobile phone. We also have a server-side version for people that want to have a standalone version working in a secure environment, or to be able to work with large data sets, having some stronger hardware underneath it. So actually all you need to run the tool are the locations of your mRNA spots and some sort of data frame that represents cell type specific gene expression. Over here, I've actually recorded SAM Lite, on being run on a mobile phone. So I've just gone to the website. We see the landing screen, and we can go to the data center and start uploading some coordinates for gene expression. So this is an in situ sequencing data set of a human pancreas of around 300,000 spots. So these are loaded within a couple of seconds in real time, and you can zoom in and view the tissue and see where these molecules are. We can then upload a gene expression signature matrix, where we see how gene expression corresponds to these cell types. The tool itself only has 3 parameters, so it's not so many, and most of these run fairly easily. I'll just set this to some reasonable parameters just for the sake of the demonstration. We then get to the point of running the kernel density estimation. So this is the slowest step of the entire analysis, and for this small data set, it only took a couple of seconds. And then we can annotate the cell types. So again, in a couple of seconds, we're able to generate a cell type map of the pancreas tissue, where we can interactively zoom in and see cell type distribution within the tissue. So these are all interactive plots where you can download zoom in sections, reset and pan the image as you like for making some reasonable figures for reports and for publication. So the major selling point here is we were able to actually analyze a data set within 2 minutes without a bioinformatician, without some heavy compute hardware. So this was really just done on my mobile phone. So it changes the way we go about thinking how spatial data can be analyzed. All right, so the last part of the talk, I'm going to talk about Plankton.py. So this is the tool that will be used for the tutorial after this talk. And this is a framework for analyzing spatial relationships between molecules from single molecule spatial omics data. So the previous tools had very specific goals in mind, whereas Plankton kind of takes this more exploratory data analysis approach to providing you an ecosystem where you can actually analyze single molecule data. Again, it builds on this this idea that there are a lot of tools and statistics for point data for spatial omics. And we made the tool freely available on Plankton.py. So I think the current version that's available on GitHub corresponds directly to the tutorial. And it has a simple Pythonic data handling concept, and it's very lightweight, and most of the development and analysis takes place on a laptop. And the entire paradigm focuses really on this kind of exploratory data analysis framework. So you would typically run this through some sort of Jupyter notebook. It interfaces into the existing ecosystem of tools quite well. So the input format is decoded spot from the Starfish pipeline, but any sort of matrix which defines the gene and X and Y location can be easily converted into this decoded spot format. And then it interfaces with ScanPy for some QC and cell type analysis. And we can also export the Plankton.py data objects into SquidPy for downstream cell-based analysis. In terms of data handling, Plankton.py introduces a new data structure called an S data object, a spatial data object, which is very analogous to the AND data object used in ScanPy. So if you're familiar with that, a lot of these S data object functions will be really intuitive. So they're all based around this pandas data frame functionality. And a lot of the functions built in allow for easy data manipulation, visualization, and some easy analysis. So Plankton.py leverages a very large number of statistical tools in the Python universe. It inherits pandas analysis functions, so things such as simple correlation analysis. It's compatible with SquidPy's spatial statistical framework, such as neighborhood enrichment analysis, so you can see how cells are co-associating with each other, or sorry, how genes are co-associating with each other. It integrates a lot of the SciPy and scikit-learn functionality, so you can really do some efficient computation and machine learning, such as nearest neighbor analysis. And it has some properly implemented specific spatial statistical functions, such as co-occurrence analysis, as shown in this particular figure here, and you'll learn a bit more about that within the labs. One of the things that makes Plankton.py really useful is its visualization tools, and it provides a lot of plotting functionality and interactive analysis. And this is based around pyplot scatter function, so it allows you to do easy integration of spot-based and grid-based data analysis. You can also integrate things such as background images. So if you have a DAPI stain, you can have this as an underlay for your images. And when it comes to interactive analysis, we can also circle regions of interest within our lowly represented dimensional data within web interfaces and Jupyter. We also have a type binding to single cell data. So this can be required for annotation of cell types if you already know what the cell type signatures are. We can also do statistical comparisons with molecule distribution, and then also perform supervised cell typing analysis using an adapted interface to the SAM algorithm. All right, so moving on from the features to actually describe what Plankton.py is doing in terms of its modeling approach. The local neighborhood modeling approach is very similar to what's adopted in SAM, and it performs a radial distance-based modeling. So for each molecule within our data set, we generate some sort of gene expression vector that is built up of a local neighborhood graph that's weighted by a particular Gaussian. Once we have these local graphical models for each of the individual molecules, we can then represent this using a UMAP algorithm, which is regularized in a custom way to optimize it to reveal intricacies about the spatial context. To show this in a demonstration on the mouse hippocampus data, so this was developed, this was generated from Mats Nielsen's group using in situ sequencing a couple of years ago. Over here, we have an image of the individual molecules detected from the spatial transcriptomics experiment in the mouse brain. We apply Plankton.py, so this looks at the neighborhood of each one of these molecules and builds a neighborhood graph and represents it in a UMAP. It's quite nice. We have a highly structured pattern here. So we have these nice distinct clusters, and we have this really long elongated continuous cluster as well. When we map back these colors to the tissue, we can actually see that they correspond to discrete and continuous tissue regions, where you have very discrete layers within the tissue, and also this continuous gradient, which actually is not so well described in the context of the CA1. So we were very excited to see that this was discovered de novo within the tissue. We can also use it for analyzing other data sets. Over here, it's applied to the human pancreas. So what we do here is, again, we take the individual spots, we apply Plankton.py, and we get a UMAP representation of the tissue context. And by looking at where these genes are expressed, you can immediately see that there are very localized gene expression patterns of that can be isolated, such as the exocrine tissue. This part of the UMAP, we have the islets, and at the bottom we have the main draining duct. We can also go into more detail. So in this region with the islets, we can also look into the substructures and break this down into the alpha cells and the beta and delta cells within it. So this is really an excellent exploratory data analysis tool. In this particular data set, we found that there was a region of genes that were being expressed that didn't really correspond to a cell type. And when we isolated that on the tissue, we found that this corresponded to background noise. So this was a very exciting use case of the tool where you can actually use it for background noise filtering. So by removing the signal associated with this part of the UMAP, we can then filter it and have a much cleaner signal for downstream analysis or for visualization. So hopefully with that, you have an idea of how we can model spatial relationships between molecules using Plankton.py. And it allows you to easily explore your data interactively in ways that other tools really cannot. Because I think right now, like this idea of exploratory data analysis at the molecule level has by and large been ignored. And it's a very important space to explore because once you get to segmentation-based results, you lose a lot of your information. So actually having segmentation-free analysis before you get into segmentation-based analysis is quite important to make sure you're not losing information. So using our tool, we're able to identify discrete and continuous cell types within the tissue. We can disentangle noise from foreground signal. And the tool itself is tightly integrated into the Jupyter Notebook workflow, allowing for really interactive data analysis and exploration, for building models, and for testing hypotheses and analyzing the data. All right, so as a quick summary, I've hopefully demonstrated to you that segmentation and identification of cells is not a requirement actually to make sense of single cell or single molecule spatial transcriptomics data. We were able to generate cell type maps, identify spatial relationships between cell types, and also identify tissue domains without identifying a single cell. I presented a couple of tools, Plankton.py, which modeled molecule relationships to uncover tissue structure. I talked about SAM, which successfully employs segmentation-free approaches for spatial cell typing and then identification of domains. And SAM Lite, which is our lightweight implementation of the SAM algorithm. I'm very excited about these most recent tools that we have. So Plankton.py for this unsupervised analysis, and also SAM Lite, especially with regards to how these web tools actually democratize access to computational methods. So this now allows molecular biologists that want to explore the data to be able to quickly look into it without the need of bothering a overburdened bioinformatician. It would be great for data generators to quickly troubleshoot and provide quick overviews to customers of their data, and also for data analysts that just want to compare segmentation-based results against raw gene expression data. All right, so that's the end of my talk. I'd like to make quite a lot of acknowledgments, in particular, Sebastian Thiesmeyer, who developed SAM Lite and is the developer of the Plankton.py algorithm. He helped me a lot also to make this presentation and focus on what the key take-home messages were. And also Jumping Park and Ronald Troy, who were the original developers of the SAM algorithm and inspired the idea of segmentation-free analysis within my group. So with that, I would happily take any questions that you may have. Thank you.